Good afternoon. Let me share my screen. Okay, I hope my screen is visible. Okay. Once again, good afternoon to all of the participants. The day 2 of the FDP session. Today, basically, we will focus on protein family selection and effective, efficient data extraction strategy. So, if you remember, in the last class, we basically focused on the protein sequence and structure databases. We discussed about UniProts and we discussed about protein data bank. We you have noticed that how efficiently we can use the UniProt databases and we can find out our sequences, structures, as well as various very insights parameters like uh, subcellular localizations, the isoforms, the domains, and some other binding sites as well, right? And at the same time, we have seen that in the protein structure databases, the how the data is organized, how the atom section is organized, and also at the same time we have discussed some errors that is found in the file formats of protein structures and how to manage them. Okay, so these are the main potential source of your error in you can say error in uh, docking or simulation. So this sort of errors basically we find. Okay. Okay. So let's start today's session. Now, you know about secondary databases. Last day itself, we talked about that the databases are of two types, basically primary and secondary resources. So primary resources we discussed yesterday, that is the UniProt and PDB are the classical examples of primary resources. Actually, we divide the databases into two groups depending on the data sources. In the primary databases, data is directly submitted by the researchers. So, but in secondary databases, data source is the primary databases other than researchers. Okay, so for a particular purpose, a secondary database is means compiled, and the data from the primary databases is retrieved in these cases. That is the differences. So now you see that today we will discuss some important secondary databases and their applications as well. So both the theoretical coverage and hands-on sessions uh, will go through. Okay. So now the first one of our today's topic is structural classification of protein, that is the SCOP databases. Now all of you know that proteins are nothing but the series of amino acids. That means these are the polypeptide chain where amino acid residues are joined through polypeptide peptide bond. Okay, so it has the directionalities, that is the N-terminus and C-terminus. Okay, it has the N-terminus and C-terminus end. Now, if you see that this polypeptide chain is first locally folded, the local folding you categorize it into alpha helix and beta plated sheets, and then these local folds are means folded into its three-dimensional structure. And if a protein is having more than one chain, like you have seen in PDB, that chain A, chain B, chain C, like this, then they will have the structural functional unit is the quaternary structure. Otherwise, if it is a protein is having a single chain, you know that the tertiary structure is functional one. Okay, if more than one chain, then the quaternary structure is functional. Okay, so are the levels of structure. Now we will see that how these structures are classified in the secondary database, that is SCOP. So what is SCOP? Let us discuss in a few minutes that SCOP is a manual classification. Okay, it's the classification that we will discuss now. Actually, they are manually classified. Okay, and this classification is based on similarities in their structure as well as amino acid sequences. That means this classification considers both the sequence and structures. That means the amino acid sequences as well as three-dimensional structures. Okay, so this scope classification is basically focusing the evolutionary relationship between protein. Okay, that is the key important features of this database, that how the protein has been emerged or diversified from one species to other. That is the key criteria of SOP classification. Okay, and proteins, you will see that with same shape is same, but if they are having a very poor sequence or functional similarity, they are grouped under different superfamilies. So there will be families and superfamilies concept. We will just go through it. Okay, just hold on. We will go through it. Okay. So first we are trying to understand that what is SCOP all about, what we are going to learn in SCOP. That is the first few points we are discussing now. So again, we revise. SCOP is the manual classification of protein structured domains, right? And this classification is based on the similarity in 2D and 3D structures as well. And this classification focusing the evolutionary relationship of protein. So that's why many times it becomes very critical in our research to find out the evolutionary relationship among the proteins we are interested in. So that in that cases, this structure is going to be very helpful. So now proteins, as I said, that superfamilies, the superfamilies contains the proteins where shapes are similar, but very poor sequence and functional similarities. At the same time, if you have same shape and if you have improved similarity level, both in sequence and functional levels, then they are grouped under families. Okay, that means from superfamily to family. That means under a superfamily, there can be number of families. Okay, under a superfamily, there can be number of families. And we will assume that these proteins who have the common families, they will have a common ancestor. So they will have a closer common ancestor. And this SCOP database was created in 1994, you know, and it is maintained by the Center of Protein Engineering, CPE, and the Laboratory of Molecular Biology, that is LMB. Okay, and it was maintained by Alexei G. Morgine and his colleagues at CPE and LMB lab in England. Okay, so these are the hierarchy we are talking about. Okay, so at the top there is class. At the top there is class. Now one class can have many folds. One class can have many folds. Now one fold can have many superfamilies. Now under a superfamily there will be many families, and number of protein domains constitute a family. Number of protein domains constitute a family. And these different domains you will find in several species. So several species will share a common domain. And that is the beauty, that domains are found in functionally similar organisms. Okay. So once again, it is the class, fold, superfamily, family, protein domain, and species. So now you see that SCOP classifies protein structures into hierarchical level based on structural and evolution relationship, right? Just we have talked about this evolution relationship. And these classes have broader structural categories like all alpha, all beta, alpha by beta, and alpha plus beta. We will explain all these categories. Okay, we will explain all these categories. Then we have folds, just we have seen the hierarchy folds. So domains within a class that share similar core structures or topology. That means connectivity of the core domains into a fold will differ from one to other with respect to topology. Topology is nothing but the geometry of connectivity of protein. And you know that this geometry plays a pivotal role in function determination. Okay. So in superfamily, you will see that there will be many of such folds and they will have a distant common ancestors. Okay, and they will have structural homologs. In family, you know they will be very closely related, and a protein domains are very unique structures. The domains are very, very important one. You know that the region in the protein structure that can determine its own folding and function, that part is termed as domain. And even if you detach this region from the rest of the protein, it can retain its function. That is the main importance of this domain. That's why we are interested in domains, because domains can be shared among several proteins, and these several proteins will have multiple functions, and this domain will be equated by their functions. So, and species and domain, you now know about this domain is different from protein domain. Please note that this is a distinct structure and functional unit of a protein. For some proteins, it may be the entire structures. Now, these are the options we are talking about. So you see that if we go through structure based or evolution based, if you see from bottom to top, if we go to bottom to top, then it is termed as structure based classification. If we are coming from top to bottom, then we call it evolution based classification. Now, as I said earlier, that pop has several class. It starts from the class. Okay. Now you see that there is actually four classes. COP has basically four classes. There are some several other classes we will see. Okay, some minor classes are there. All total 11 classes are there, but majorly there are four classes. Okay, what are these four classes? Alpha, beta, alpha by beta, and alpha plus beta. What do we mean by this? This alpha fold class alpha indicates that in this class of proteins, all the proteins will have alpha helix only as secondary structural unit in their three-dimensional structure. Okay, so all the protein will have only alpha structure. Okay, are you getting my point? Just a minute. Just a minute. Okay, so alpha means all the structure will have the similar class, that is alpha. And similarly, beta class indicates that in this group of protein, there will be all the secondary structures of beta type only, beta sheets, right? So in one class, we have got alpha, and one class we have got beta. Okay. Now the confusion comes that what do we mean by this alpha by beta or alpha plus beta? Okay. So alpha by beta actually indicates that this group of proteins will have both alpha and beta type structure. Now, what is this group? This group also indicates the both alpha and beta type of structures are present. Then you will ask me, then what is the difference? Okay, then what is the difference? Both the structures are having alpha and beta. The difference is in their topology. Okay, difference is in their topology. So let's us revise once again these four structural classes. These are very, very important, and this eventually influences their functional determination. Okay, so we have one group of protein, one class of protein. In this class, all the proteins you will find they will have only single type of structural secondary structural unit, that is of helix type. Okay, alpha helix. Now in second group of protein, there is again only one secondary structural unit, that is beta sheet. You know that we have two secondary structures. Just in the beginning, we revised briefly, very briefly, that we have local folding two types. That those are alpha and beta. Now in third and fourth group, this alpha and beta is combined, but their topology is different. That's why one class is called alpha by beta and the other class is called alpha plus beta. Now, what is the difference? That is the point. So, you see that if it is alpha by beta, you will see that all alpha and all beta will form a separate layer. Okay, so that is the topology, that here all the alpha that is present in this structural type, they will form a particular layer of alpha, and other one, they will form a particular layer of beta. So that is alpha by beta. And in alpha plus beta, you will see that alpha and beta is mixed. That means one alpha will be there, and the alpha will be followed by a beta sheet. Then again, it will be followed by an alpha, and it will be linked through some coils. Okay, so these are the differences in between these classes. Now, say if we go in the evolutionary based classifications, you see that so after the class, after one class, there will be many folds. So after a class, there will be many folds. So you see that folds, if we consider only alpha by beta, they have three different types of folds. Okay, what are the folds? See, Rosman folds, flavodoxin-like folds, then alpha by beta barrels. Okay, in this way, under class alpha, alpha by beta, sorry, there will be several folds. Now, see, under the folds, there will be several superfamilies. So now you see that an example is here, that is alpha by beta barrel. This is one class of fold, one type of fold. Sorry, we should not use the word class here. It is that fold type. So alpha by beta barrel fold. Here you see there are so many superfamilies. That is TIM superfamilies, strip biosynthesis superfamilies, okay, glycosyl transferase superfamilies, Rubisco superfamilies, okay. So these are the superfamilies, and earlier we have discussed that superfamilies, they share common shape, right? They share common shape, but their sequence similarity among the proteins will be very poor, very poor. Say less than 30% sequence similarity will be there. So this superfamily will share very poor sequence similarity. Now under a superfamily, there will be several families. Under a superfamily, there is several families. Like here in the example, you see glycosyl transferase is a superfamily. Now, under the glycosyl transferase superfamily, you will find beta galactosidase, beta glucanase, alpha amylase, and beta amylase. These are the family. Now, as per the definition of family, earlier we discussed that they will share common shape as like the superfamily, as well as they will have high protein similarity also, right? So now you see that how we are traveling. We have class alpha by beta, then we have Rasban folds, alpha by beta barrel, then alpha by beta barrel has several superfamilies. Now, glycosyl transferase will have several families. That means beta amylase is a family of proteins. So under this family, there will be many proteins. Under this beta amylase family, there will be many proteins. So this beta amylase family, it belongs to the superfamily glycosyl transferase. And this beta amylase family belongs to fold alpha by beta barrel fold. That means we know that in beta amylase, there will be alpha and beta structures, secondary structures, and this alpha and beta will be spatially surrounded each other and form a layer of alpha helix and a layer of beta sheets. And this family, these alpha amylase belong to the class alpha by beta. Okay. Now, if we talk about that under a family, there will be several protein. Now you see that under alpha amylase, okay, there are so many proteins. They forms under alpha amylase. Okay. So under alpha amylase, there are so many proteins. Right. Now the species. Okay. Now the species. We are talking about the species, right? We said that a protein can be found in many species. Okay, so you see that this protein, this glycosyl transferase, okay, can be found in several species. Okay, several species, and they will have the PDB ID. These are the PDB ID of their structures. Okay, that is present in the databases. So what we have learned so far, so what we have discussed so far, we have revised our means basics idea of protein classification. Okay, and here to understand the protein, its structure and function, we have classified the protein in such a way that very easily, like we have classified the organisms, so that we can study them in a scientific way, right? The taxonomical classification of all the organisms, the plant, the animal, and all these five kingdom classification. It's similar. We have classified the proteins into class, fold, superfamily, family, protein, and species, so that we can understand its structure without going in details about that particular protein. So if we know that this particular protein belongs to this family or this superfamily or this fold, then we can come to know what is their structure and how they actually function, their mechanism of function. So these are the main classification of form. If you have any doubt in this point, you can ask me. Okay. So now you see that this is the database, how we can access the class scope database. Just within few minutes, I'll show you going through the database. Okay. So you see that these are the home page of scope databases and these are the classes. Now that's what I was talking about, that four important classes These are the four important classes, and there are some other classes. Okay, and out of these other classes, you see that these are not a true class. These are not a true class. Okay, so there are some small classes of multi-domain proteins and membrane and cell surface proteins. Otherwise, all the proteins mostly they are classified into these four different classes. Okay, so these are the 11 different classes. You can get to know that that classes. Now, let us see the structure and understand. So this is, I hope you can understand looking at this protein, what class it belongs to. You are right. You are absolutely right. These are the all alpha proteins, right? These are the all alpha proteins. You see that in its structure, there is alpha helix only. No beta sheet is present. Can you see? No beta sheet is present here. So this is a bromodomain protein. In this bromodomain protein, it's a globin fold, okay, and this one is hemodomain fold. Now you see the beta propylar protein, okay, it's a all beta protein. You see, just look into its structure very critically. You see there will be no alpha helix, okay, and immunoglobulin domain. Just look into this structure. Okay, it you might be misguided. These are the these are the helix, but these are not. These are some coiled region. Okay, these are some small coiled regions. They are not at all helix. So you see that HH3 domain, the beta propylar domain, immunoglobulin fold, they are all belongs to all beta protein. That means distinctly you can see that the all beta and all alpha. In all alpha, there is presence of no beta sheets. In all beta, there will be presence of no alpha helix. Okay. Now you see that the alpha by beta. Okay, the tin barrel we are talking about, or say leucine rich repeat. Okay, leucine rich repeat, the flavonoxin fold. So many times in the structure database you actually came across this sort of names. Now you can see that these are the helix, okay, and these are the sheets, and see their topology. Look at their topology in both the structures. Okay, so these are the secondary structures, and these are the alpha helix, the structure, and these are the sheet structures. See, they have formed a different layer altogether. Okay, they have formed a different layer altogether. Okay, now you see that these structures, these are alpha plus beta. Now, can you notice this one? See, this is a helix, right? So now you see where its origin. Can you notice this one? This is a beta sheet. Just let me show you. This is a beta sheet, and this is a coil region connecting this beta sheet. Okay, and this is a beta sheet. Okay, and it's connecting this helix. You see, it's connecting this helix, and this helix is again connecting this beta sheet. Okay. So now we know that all the alpha helix and beta sheets, they are in every. So now you see there are so many structures, and these are the additional classes. So if you are interested, you can go through in details about these additional classes. So from the Google, we can uh, explore this databases. So in the scope, there are two versions, two prototypes, scope 1 and scope 2. The organization is different only, so we can get any one. So next we will discuss ProSite, but because earlier of this means before going to ProSite, databases, we will see the SCOP databases. Okay, so let's go to the SCOP database. SCOP database. Okay, so this is the structural classification of protein. So I hope now you can see this one. So as I said, we have two prototypes. Either we can go through scope 1.75 or scope 2 prototype. So if I go through scope 1.75 prototype, you can see these are direct just some folders that you can get through, but we can search it from here, the prototype 2. Okay, so now you see that these are the search area. Okay, these are the search area. So how we can search this protein? Now you can see the statistics. Okay, this is the latest updates. How many proteins are there and how many structural folds are there. So you can go through the statistics. Now you see that we can search by text or scope ID, PDB ID or UniProt ID, whichever we are having. So if we are doing some research, we might have some PDB ID, we might have some UniProt ID. So last class, in last session, we discussed about the UniProt databases. So I hope you are familiar with UniProt ID. So now say we just for the first time we are going through scope. So I might have some PDB ID. So see the PDB ID to AJF. See, this is a PDB ID. Now, if I see go, see, it is searching my protein. Okay, so it's this. There is the result. Okay, so you see, I have two domains. Okay, it has two domains into two AJF. In this is the chain A. Okay, this is the chain A, and we have two domains. Let's see. See this one. So this is the what is the source species? Source species is the Homo sapiens. Okay. If I am interested in ancestry, so I will get the ancestry. So this is the class. Okay. So each class is assigned a number. Okay. So this is the alpha and beta protein. This is alpha plus beta, right? So when we see that this protein belongs to alpha plus beta, immediately it comes to our mind that what type of structure is this. Now, what is the fold? See, this is the ginseng-like fold. Now, what is the superfamily? So, this is the superfamily. Now, this is the family and this is its domain. Okay. Now, you see the domain. So, these are the positions of sequences. You can see this is marked. Okay. So, this is the domain region. So, this is the whole sequence of this protein. This is the sequence of 2 AJF. Okay, this is the sequence of two AJF, and this is the domain. Okay, now you know the significance of domain. We already discussed the domain structure, so they are very important for function. They can retain its function, they can fold independently with respect to the other part of the sequences, and they can have the 3D structures without the influence of the rest of the regions. Okay, so these are the domain structure. Okay, now, so then let's uh, search another protein, the hexokinase. hexokinase, a text search. Now we are doing the text search. So let's see. You see that we have got one families. Okay, now under this families you have seen that seven domains are there. Okay, seven domains are there. So these are the regions of domain. That means the position of sequences. Okay, and these are the ID. The ID actually, by looking at this ID, you can see that this is the protein data bank ID, and this is the ID of UniProt. So from here we can get the detailed structure of sequence, details of UniProt. So you can open this one. You see these are the details of sequences. Okay, right? These are the UniProt databases. So from UniProt, you will get the details of these sequences. Okay, so this is the human hexokinase. So this is the length of the amino acid. Just yesterday we discussed this right now. This is the annotation code. Okay, this is the function of this protein. Okay, these are the structures. Okay, these are the structures, PDB structures, available structures. We discussed about this one, right? Yesterday. So we will not go in details of this one. So from here we can get this one. Now you will see that hexokinase 4, hexokinase 1, this is an hexokinase 1. So these are the different domains we have found in different organisms. You see that organism is different, right? So, and you can get the details of its information from this one. Okay, so there is a different family. So if you go to the ancestry, you see these are the ancestry means this hexokinase belongs to this class. Alpha, again, it is a protein that is alpha by beta, right? So now you can differentiate the earlier one and this one are different. So topologically, they are different. Both the protein will have alpha helix and beta sheet regions, but topologically, if you consider, they are going to be different one, right? So they are having this super family acting like ATPases, the family hexokinase. Okay, so hexokinase is a family of protein. So hexokinase is not a single protein; it's a family of protein. So if you go back to the structural hierarchy, you see that under the family there are so many classes. Now you see this is the family. Okay, so under the family there will be so many proteins, and one protein will be present in several species. I hope this is clear to us right now. So one protein can be present in several species. Now see, we are interested in these proteins that how they have evolved, if how mutation has been accumulated, and what are the means mutation accumulation pattern across the species, and from there we can also analyze the species diversifications also. So that is the different way of research. Okay, so this is all about scope classification. If you have any questions, you can ask me in scope classification. Okay, so if not, then we'll move to next slides, that is ProSite. Okay, so ProSite is a database of protein families of and domains. Okay, now it is another secondary resource, that is ProSite. But you may ask that, sir, then why there are so many resources? Okay, so you will see when we'll go through the ProSite, you will see the application of ProSite is bit different to that of SCOP. In SCOP, if you have a protein, you can analyze that in which family they are matching. But in ProSite, if you have a sequence, okay, then you can scan that in with which known proteins your sequence is having some functional similarity with the domains or not. And it was developed by Amos Bayrock, okay, in just a minute. It was developed by Amos Barak. And if you see the current release of this Proside as per 2025 January, okay, and this is the last release 2025-01, and the 5th February. It contains 1952 documentation has got entries, 1311 patterns are there, okay, and 1399 profiles are there. Okay, so these was means maintained by Switch Institute of Bioinformatics and the EMBL. Okay, so Sweden and European Union, so both are funding this project. Now here actually we are interested in some motifs and domains. Those are having a non-randomness. Okay, those are having non-randomness, and they are conserved across the species. So that is our importance. So protein domains and motifs many times we actually apply them as synonyms, but they are not, right? You know that the domains and motifs, they have differences. If you go for the domain, you will see that the domains are basically 50 to 300 amino acids long regions. Okay, the domains basically 50 to 300 amino acids long, whereas the motifs are basically 10 to 20 nucleotide amino acids long regions. So basically, motifs can be part of domain. Okay, so domain actually represents that these are the regions of sequence which can fold independently, which can retain its function. As I said earlier, even if you detach these regions from the rest of the sequences, but motif is not of that type. Motif's function will be compromised if you change in the sequences of the flanking regions, right? So that's why motif and domains are quite different. And if you see the key differences in between motif and domain, you can see that they are largely independently folding structural unit, and they are short conserved sequences. Motifs are short conserved sequences. As I said, just see that they are the larger units and they are the smaller units. And stability, if you talk about stability, they can fold and function independently, but the motifs, they cannot fold or function independently. And functionality, if you talk about functionality, they are having a distinct biological function. Like an example, if you see the kinase domain, okay, HH2 domain or immunoglobulin domain. Okay. On the other hand, if you see the motifs, usually they serve as a functional site. Okay. Usually they serve as a functional site within a domain. Okay, like say zinc finger motif. Okay, helix turn helix motif and Walker motif. So these are the examples of some motif-like structures. Okay, these are the structure of some motif-like structures. Okay, so now we will see that how actually they are designed or they are searched, how motifs are actually positioned. Okay, so there is a concept called profile. What is a profile? The profile actually it is a matrix. Matrix value. You know that matrix are some rows and columns, right? So the columns and rows will represent the sequences, sequence one and sequence two. If you put row wise and column wise, then across the matrix cells, you can get their values position wise, right? So it's a matrix. Basically, it's a weight matrix. Weight matrix means it will give you some values. Now, what is the application of profile in bioinformatics? Just we have talked about domain, right? So, and we discussed that the domains are remains conserved across the species. Obviously, the domains have their distinct functions, so they remain conserved. But are they 100% conserved? Of course, they are conserved, but sometimes they may be slightly different. Okay, and this difference can be positive or sometimes very rare it can be negative. So positive and neutral mostly same. Now, if you get the domain from say 10 different species, now you align these 10 different species position wise, column wise, then you will see that at a particular position for the timing you assume that alanine is there. So that means at that position alanine is accepted. Okay, other than alanine, that means there are 19 other amino acids, so those are not actually accepted. So this profile is a 20 by 20 matrix. They will give you the values for a particular position for all the amino acids. That means here in this cases, the frequency of alanine, that is 100% frequency in this cases, that will be converted into a weight value. Say 10 and others will get 0. So in this way, weight value is provided. Okay, so let us see an example. See, this is the sequences. There are sequences. There are 7 sequences. Now, in a particular column, you can see what is the frequency of F, what is the frequency of Y, what are the frequency of L, and rest of the residues frequency is 0. So in this way, just considering their biological structures also, you will put some values. So column wise, so this value indicates this column, column 1. This value indicates column 2, 3, 4 accordingly. So for these sequences, you are actually converting this sequence into a profile matrix. So many times what we do, if we use a particular sequence for a search, so you think that you have a sequence of kinase activity. Okay, you have a known sequence of kinase activity. Now what you want to know? You want to search the database that what are the other proteins having kinase activity. Now, what actually we do? We use our sequence that is known to us as having kinase activity. What we do? We means use this sequence as input. That means actually you are getting bias to your query sequence, right? So your query sequence is not the representative one of the kinase activity proteins. So if you are using your protein only as input, that means you are biased to your sequence, and because of that, you might miss some other actually true positives. Those are having kinase positive, but your program, your algorithm will be failing to retrieve that. So that's why instead of using a particular sequence, many times we use profiles as input. So the profile actually, you can assume that in your class, you have a particular class and you say that what is the performance of the class? So if you take any one student and if you say that his or her performance will be treated as the performance of the class, where you are going wrong, you are getting biased to that student. If that student is doing well, you will say that yes, the class is doing well. If that student is not doing well, you will say no, the class is not doing well. But see where you are going wrong. But instead of that, if you take the average performance of the students and then you compare your department with the department of other universities or other colleges, I'm not asking you to compare, just for the example purpose of things, I'm saying that then you can say that the biotechnology department in this university X and biotech department performance of biotech department in this university Y. You can compare, but you cannot compare one students from this X university and one students from that Y university. Then you are getting biased to those students. So that is the wholehearted objectives of these profile values. Okay. So there are so many applications of profiles, the conserved regions, variable regions, and patterns and domains identifications. They are done by this um, patterns. So now we will see this is a consensus sequence. This is another concept. How consensus sequences? So there is A, H, and T. So if you consider just 3 to 4 sequences, it will be very difficult to say that how we can derive a consensus sequence. But if you have many sequences like this one, you see, now you are in a position that what will be, see your consensus sequence. So as A is maximum in these cases, so there will be many other sequences also. So you might consider A in this position. Okay, in this sequence, A in this position, T in this position. So by applying many sequences, say 50 to 100 sequences, you might get. So although there is A, you can see that logo, the conservation is saying that it is the low conservation, right? As you can notice that A is there only at few sequences, only A is there. So it's a low conservation. But if in these cases you see in all the sequences there is T, right? So you see that high conservation. So this is the logo. Okay, logo is explaining the conservedness. So in this way, we can prepare a consensus sequence. So now, actually, what is the objective of means these slides to explain that how we read this one, how we read this profile. Okay, this profile, this consensus sequence actually means that at position number one there will be A. At position number two there will be T. At position number three there will be H. But at position number four there will be either D or E, and chance is 50%. Okay, 50% chance there will be D and 50% chance there will be E. That means if I am using this sequence to search my similar sequences, then I am getting bias to this sequence. And similarly for this one and this one. But instead of these three, if I prepare a consensus sequence and instead of this one, this three, if I use this consensus sequence as query, then my query sequence is not biased to a particular sequence. And this will be a better representative like the class average I talked about, and it will give you the maximum chance of getting the similar sequences. Okay, so these are the consensus sequence. So now I will tell you some other parameters. So there are these are the standard IUPAC 1 letter codes for the amino acids we will use. Okay, the symbol X is used for a position where many amino acids is accepted. So simply I will explain you that how it is actually read, how the pattern is read. Okay, so let's all these things can be explained here in this sequence only. So let's see how we can interpret this one. Okay, so here at position one, you see now you understand that there will be either A or C. Okay, either A or C at position one. At position two, it is not conserved. The position 2 is not conserved. Many amino acids can be accepted at this position. Again, the third position is V. Valine will be at the third position. Now, at the fourth position, again, it is not conserved. Not only the fourth position, four consecutive positions. Okay, from here, four consecutive positions will have a non-conserved regions. Are you getting the point? So I repeat once again, at position number one, there will be either A or C. At position number two is not considered. Position number three is valid. And position number four and next four residues, means next three, total length is four, will be non-conserved regions. Okay. Now, what next? What this indicates? The curly bracket indicates that in this position, there can be any amino acids except E and D. Okay, this curly bracket explain that at this particular position, there can be any amino acids except E and D. Okay, so these are the protein. Okay, so let's see. Uh, just let's discuss the ProSite database. Let's explain this one. So to scan the ProSite database, we can go to web and we can see ProSite database. We can access ProSite database. You can see this is listed at the Express server. Okay, protein. So this is the ProSite database. That means you can express your patterns. Okay. Now here you can see if you have the means some pattern, you can search the pattern. You can browse by documentation entry. Okay, so there are so many things. Okay, so these are the domains, images, how it's designed the domains. Now say here we are going to see that if there is any patterns or not in my sequences. So say I have got some sequences from databases. So let's see. So I have already downloaded some sequences. This is the albumin protein. Okay. So this is the albumin protein. So this is one protein that is albumin. Now see, I am taking this. So this is human albumin protein, right? Human albumin protein. So I have already taken this protein from the UniProt databases. Now, see, I want to search some patterns here. So if I say, okay, you can see, the pattern. Now you can see it's so accurate, right? So these are the domain. So these are the see, it has found the domain. This is second domain and this is third domain. So it has predicted three domains. Okay, and this is the scale. You can see the ruler. So this is 1 to 1000. You know the sequence is 609 amino acids long, and the 60 amino acids long. From which position to which position are the sequences? You can see this one. So this is domain number one. This is albumin domain. See, these are albumin domain. It's indicating the albumin domain. So that means in my sequence, in my human albumin sequence, it has three different domains. Okay, and see, can you see these symbols? See, these symbols indicates there are some disulfide bond. Can you see these bonds? These disulfide bonds. You cannot see this one because it's a protein, it's not an enzyme. So if you are using some enzymes, you will see, you will get the active site of your enzymes. You will get the active site of your enzyme. Okay, and these are some other sites, other ranges. Now, if you go in details of this domain, so it will show you the prediction score. Okay, so this is from 19 to 201 position, right? 19 to 201 position. So this is the position of disulfide. Okay, so amino acids at position number 99 and amino acids at position number 115, okay, will form disulfide bond. Okay, you can see this one. These are the disulfide bond. So at 114, okay, at 114 and 125, there will be another disulfide bond. Now at 148 and 193, there will be another disulfide bond. Okay, so you can count this one. See, this is domain 2. So many disulfide bonds are there. Okay, so you can do the study in details. Okay, fine. I hope you are getting the point, right? So if there were any active sites and other domains, so we could have. Okay, sorry. Okay, so these are the domains. I hope you have got this one. Any questions at this point? Okay, so we are done with society databases. So now we will move to CAT databases. Okay, so now you know that CAT is bit similar like scope, but there will be again some differences in the data organization. We will see that. Okay, so you see that the CAT database provides again an hierarchical classifications. Okay, but the hierarchy is different in this case. So we'll see that. Okay, and these domains are obtained from protein structures deposited in the protein data bank. And the data is both manual as well as automated computing. So in case of SCOPE, we found that all the entries are manually reviewed, right? So if we see that what are protein families, the protein family is a group of protein that share common evolutionary origin reflected by the related functions and similarities in sequence and structure level, right? So now if we see the difference in scope and gap, so we otherwise we might have the question, right? That scope, if I have we have gone through scope, then why we go need to go through uh, CAD? So here you see that the class architecture, topology, and homology. That is the hierarchical classification. So it is the manually curated classification. It is semi-automated classification of proteins, right? Then what is its scope? Its scope focuses on evolution relationship. Scope focuses basically on evolutionary relationship. Okay, and accordingly they form the hierarchy. But the CAT database, they use the combination of manual and automatic classification into architecture and homology and symmetry. Means they are targeting the functions. They are not just focusing the evolution relationship. So they are actually focusing on function. So the primarily they are manual creation and they are automated creation. And why they are giving emphasis, you see, and they are giving emphasis in structural similarities and functional relationship. Okay, and if you see that the updates, they are more regular compared to scope. Okay, and the strength, they are having higher accuracy and they are even better flexibility. So if we are interested in designing the function and structure, then we would opt for CAT. If we are uh, interested in evolutionary relationship as well as the domain and its function, we will go for SCOP. Okay, so these are the classification of cath databases. Okay, here you can see there are three classes. It is alpha, beta, and alpha and beta. And as per the classes, they are having several architectures. Architectures are the team barrel is a architecture, Sandwich again is an architecture, row is an again architecture. And within this architecture, what is the topology? So they will have different topology. And as per the topology, they will be homologous of each other. So that is the class, architecture, topology, and homologous superfamily of structures. Okay. So these are the again the structural level with some examples. Okay, these are the examples. So the cat will provide you number of solved structures. Okay, because the entry of PDB is going to increase and you will get huge number of cat databases. Now, if you see that the database construction, it was retrieved from the PDB files and the structures that was considered 4 angstrom. The resolution was 4 and below the 4. So just yesterday I said that lower the magnitude of the resolution, higher the quality of the data. Like say if I have 2.30 and one resolution is 3.20. So which one is better? So obviously the structure, the first one, that is 2.30 is better. So we say that this one is having higher resolution. Higher resolution does not mean that the higher magnitude of this resolution value. Higher resolution means lower magnitude of the resolution value. Okay. So now this is the homepage of cat databases. This is the homepage of cat databases. I will show you this cat databases. So these are the classes, these are the classifications. So let's see uh, directly. Let's see the how we can access the database. So if we need to access the database, we can write the cat databases. Okay, so these are the homepage of cat databases. See, this is the cat databases. You can see that 151 million protein domains are classified into 6,573 superfamilies only, right? You can see. Okay? So these are the latest version of protein family. So now here you can see that you can get out the 3D structures. Okay, you can get the protein evolution relationships. You can predict protein functions. Okay, you can predict conserved sites also. Okay, so these are the different windows. Okay, like say here we can search again by PDB code or geoterms or keyword. So let's see the same protein we use to AJF. Let's find by PDB ID. Let's see what we can get. Okay, so now you see that we have got matching here in the cat superfamilies. So if we see that view all images, see these are the images of protein coding. Okay, all the proteins from this one. So now if we search through some keyword, say hexokinase, hexokinase. Okay, okay. Wait just a minute. Now you can see. Now you can see the structures. Okay, it has been loaded. Okay, so what? How we can interpret? You can see this is the geodiversity. Okay, this is the geodiversity. This is the enzyme number, EC number. Okay, diversity. These are the species diversity. Yet it has to be loaded. Okay, so they are having so many superfamilies, functional families. You can see, and these are the sequence diversity. Okay, these are the sequence diversity. Now there are 23 domains in hexokinase. We have 23 domains. Okay, these are the alignment cluster information. So if we go to the superfamily, see these are the superfamily. So these figures actually you can give in your manuscripts also. Okay, then functional domains. These are the functional domains. You can see these are the different domains of regions. The structure is mainly alpha. You see these are mainly alpha type of structure. Okay, all the amino acids. You move the cursor, you will get the chain and its position of amino acids. Helix. Sorry, this is helix. Okay, this is turns and coils. Okay, this is alpha helix. Okay, so these are the topologies. They have 404 topologies. These are the super families. Okay, and we can go through the PDB structures from here. You can see the PDB structures from here. See the structures. I hope you can see the structure. Right. So these structures, the PDB file, we can visualize that by via Discovery Studio. Here you can see this is the helix. Okay. See, this helix is joined, two helix is joined through the beta sheets. And this is again another beta sheets. This is a beta sheet. Okay. So if we view hierarchy, so these are the chain. You can see these are the amino acids. So this is the amino acids. Okay. So these are the amino acids. See the marking, it's changed, right? See, these are the amino acid number 301. See, these are the amino acid, right? You can see this one. In this way, okay, you can see this one. Now it's clearly visible, right? So lots of means analysis we can see. So we can show a 2D diagram. You can see that this is the 2D diagram. From here, we can get the ligands, where it is bound, and what are the interactions. Okay, which amino acids is bound? This we can see here, right? So this is can exit from it. Okay, so these are the now if I go for functional families, so you see there are four functional families. Okay, there are four functional families. So these are the functional families and structural neighborhood. So these are the structural neighborhood. Okay. Now the next one, if we have a sequence, say you have a protein sequence, you have found a protein and you have sequenced it in a case of disease. So you have sequenced that one. So now you are interested to find some motifs. So how we can get that, right? So for this one, we can use a meme suit. Okay, multiple expectations, maximizations for motif elicitation. Okay, so this one from this site, we can access the meme suit. It can analyze both the protein and DNA sequences. Okay, so let's see one such protein site. So if we go for meme suit, you see this is the meme suit. Okay, so this is the server of the meme suit. Now, so this one is meme suit. Now here you can see that there are meme discovery mode. We can go for classic mode, discriminative mode, or differential enrichment mode. Okay, so we can select DNA, RNA, or protein sequence. If we have a different file, so we can go for that. Okay, so now we are using protein sequences, so we will select this one. Okay, now input the primary sequences. So here you see that enter sequences in which you want to find motifs. Now you see that we have several options. We can type sequences here if we have sequences. So we can give our sequences here in this position. We can upload sequences. Okay, we can upload sequences, or you can see that we can upload bit file. So now we will upload sequences. I have the files like say mm, I want to search motif in say hexokinase. Okay, so I have several hexokinase sequences. I have 5 hexokinase sequences. I have downloaded already uh, to save time here. So you know that how to get the sequences. That's why I have already downloaded that. Now you see that hexokinase dot FASTA. Okay, so dot FASTA file has been selected. Now select the site of distribution. So I can get any sequences, occurrence per sequences, any number of this motif. So how many motifs actually I should found? I can give the number. So here I am actually finding the 6 motifs per sequences. Okay, so I may put my email ID. So it will send the result in my email ID here. Okay, I can give a description to identify the output from other entries if I am interested in. So now see, I will hit the search button. So after hitting the search button, you can see that it's showing that please wait. Your meme job has now in queue waiting. Okay, and here you will see the job details here. So I have submitted on this date, and you can see that I have got the output. Okay, and you can see that there will be several output files here, right? Okay, so if I open one output file, that is the HTML file, output HTML file, so you can see this is the motif. So as I said that there are 6 motifs we are searching for, right? So you can see this is the logo of the motif, and this is the e-value. Okay, the site, okay, and the word, that means 50 sequences wide. So these are the by default values are there. If you want to increase or decrease the size of the motif, so you can do that. Okay, so I can submit the motif to another memsuite program. Okay, or we can download this one. So here you can see the p-value of this motif. So lower the p-value, better the significance. Okay, now here, so these are the motif. Okay, so we can save this image. So this image can be saved and it can be means you can give as a figure in the diagram in your manuscript. Okay, so now you can see the positions of this motif. You can see the p-value for each of the motif. You move the cursor and you will see the motif. Okay, the start point is 214 and end position is 263. I hope you can see that. So this is the p-value. So now the p-value is lower in this cases as compared to this one. Okay, that means in this sequence one you have two motifs. Now have you observed one thing? You can see that we have the hexokinase sequence from human, bovine, horse, pig, and rat, right? And can you have you noticed that in the same position we have got the motifs. These motifs we have found at the same position. Okay, just here it's the stress size 660. Here the stress size is 662. You see more or less 6%. Why it is not exactly same? Because you see that it is from human, it is from bovine. So there is a species diversification, there is a taxonomical distance, evolutionary distance, and due to evolutionary distance, some mutation has been accumulated. Right. But broadly, you can see in all the sequences, these regions are conserved. These regions are conserved. Now you imagine that one of the sequences is unknown. Say human, this one. This is an unknown sequence. You might have sequenced this one in the lab. That means if you use this one as the query, okay, if you find the similar sequences from the databases, now if you means predict the motifs by using meme or anyone, and so that means these motifs and domains combinedly can lead to the prediction of function. Okay, and after that you can carry out one wet lab experiment to confirm your prediction. So your job is done. Okay, so we can download this one. These sequences can be downloaded. Okay, these are the sequences that has been downloaded. You can see the motifs in different sequences. You can see. So you can use this figure. So the consensus of each of this motif you can see. The consensus sequence already we have discussed earlier. What are consensus sequence? That means you can mark the color that this is the most means likely motif as per the value calculation. You can see the p-value is 0 in these cases. As I told you that lower the p-value, better the means motif. So see in these cases these are the p-value in different organisms. Now this is the um, one. Here you can see this is the consensus sequence. Now if you see for this one, this is the consensus sequence. So the motifs as well as with its consensus sequence. So now it is ready for your publication, right? So you can prepare the manuscript, you can give this one to the publication for as a figure. Okay, now we will go for let's see the XML output. Okay, let's see the XML output. See, this is the XML output. Now you see this is the XML output. So these are the positions of amino acids. Okay, so these are the output in XML. Okay, all the positions. You can see it's a huge file, right? It's a huge file. So sometimes now you might ask that it's not understandable, very difficult to understand. Then what is the purpose of it? So might be this output you need to give as input to some other softwares, and those softwares will understand the mean Excel XML format only. So that's why you can save this format, and you can this give this file as input for further means understanding or further analysis. So that's the use of this one. Okay. Now you see that we will go for mem text output. Okay, see this is the mem text output. Now you see the text output. So you will see the control sequence none. Okay, so these are the sequence and the weight, the length, sequence name, and all the details of the positions. Okay, all the details of the positions are there. Hmm, okay, so these are the motif regions. These are the sites. So already you know that these are the sites, right? In pig, in human. Okay, as per score, they have actually arranged this one. Okay, as per score. And these are the probability matrix. These are the probability matrix. Okay, here you also you can see it's a huge file. These are the probability matrix for each of the motifs. So we have 6 motifs. So we will get 6 probability matrix for this. Now see the must Excel file. You can see now here you will get the motif logo along with its consensus sequence, and here is the block diagram. Okay, this is the block diagram of the motifs. Okay, here you can see this is the logo of the motif. So actually this program, this is a different program. Okay, this program actually predicted 8 such regions as motif. Okay, and that is the probability of each of these sequences here. Okay, so the version of must 5.5.7 has been used. Now you see that the XML file like the previous one. Okay, so you know now what is the use of this one. So we'll not go through this one. Okay, now the text output. So this is the text output of the second program. Now what is the use of this one? So you can compare the output of means meme and mask. That can be compared. Okay, and which is common in both the cases, that will be more reliable. So these are the sequence they have used. Okay, we have used these sequences. Okay? So these are the output of meme suit. So if you have any sequences, you can predict its output. So if you have any questions, you can ask me. Okay, so what are the final takeaway from for day 2? So we have discussed scope, that is manually curated hierarchical classification of proteins, and we have discussed CATH secondary databases that actually determines the class architecture, topology, and homology. And also we have discussed today the ProCite secondary databases, how we can get the ProCite patterns. And also we have used MIM and MUST algorithm, that how we can detect any motifs if that is present in our sequence of interest. So that's all for today. If you have any questions, I can answer. Otherwise, I will consider this is the end of today's session.